research on combating against pneumonia.
പിന്നെ
So
ഇന്നലെ സാറേ വാപ്പ
let
മോനെ
me
ഓർമ്മിപ്പിച്ചു.
share my slides.
One second, I think.
എന്താ
One second.
കാണാൻ തോന്നുന്നു.
Yeah. Now you can go. You are not audible.
Hello, am I audible?
Yes, yes, you are audible now.
Okay. So thank you, Achisman, for your nice introduction. I am Arkadyuti from Department of Biophysics. I am learning under supervision of Dr. Krishna Kishori Nampudi. Today, I would like to share our research on combating Streptococcus pneumonia. So let me show my slides. So is my slides visible?
Yes, it is visible. You can go.
Okay. Okay. So here this pneumonia. So what is this pneumonia? Pneumonia is a lung disease mainly caused by virus or bacteria. So major cause of this disease is Streptococcus pneumonia, and it annually it kills more than millions of people. In 2009, you can see more than 2.5 million people died from this, and from this graph, you can see more the uh, patients are from immunocompromised age group, that is under 5 children and the elder ones. So this pneumonia is a gram-positive bacteria, and it causes lower respiratory tract lung infection. So let's see the symptoms of pneumonia. So you can see cough, chest pain, chills. It's more like the common cold, but mortality is very high. So let's see its treatment strategy. So for the treatment, we use antibiotics, gives analgesic antipyretic, and there also vaccination is also available here. That is pneumococcal conjugate vaccine (PCV) and pneumococcal polysaccharide vaccine (PPS). There are also supportive care like oxygen therapy. This and analgesic and antibiotics also comes under the supportive care also. You can see, but in spite of having this much of treatment strategy, we see millions of death every year. So what is the reason behind that? Let's see. So this bacteria show antibiotic resistance, and the vaccination available here. This is the major two category of vaccine, that is PCV and PPSV. That PCV is mainly for the children's one, and but the problem here is this PCV protect us from only 13 strain of pneumonia, but pneumonia has more than 90 infective serotypes, and this PPSV mainly used for adults, older ones who are aged over 65 and more than that. But this PPSV's limitation is that it is also uh, it is also useful against only 23 types of pneumonia, Streptococcus pneumonia strain. So these inactiveness of inefficacy of vaccine and antibiotic force us to find novel therapeutic approach. That's where our study comes in. So for that thing, we first see how this bacteria invade our body. So here is the pathogenesis of pneumonia. So first is transmission caused by the air or from nasal epithelium mucus, and then it colonizes in our epithelial layer. After that, it invades our host body, and then inflammatory responses come. So we thought if we can stall its infection in this colonization, then our work will be more easy. So for that thing, we see how it invades our body. So our host body is exposed to several types of diversified host pathogen. So for that thing, we have our own defense mechanism. Here I am showing our epithelial defense mechanism. So when a foreign particle comes, here this blue, yellow ones is the B cell. So when a foreign particle or foreign pathogen comes, it secretes IgA, that is immunoglobulin A. This immunoglobulin A forms dimeric immunoglobulin A here. Then this red one is the PIGR, that is polymeric immunoglobulin R, polymeric immunoglobulin receptor. When foreign pathogen comes, this DIGA binds with the PIGR, and by endocytosis it comes here, and here it goes proteolytic cleavage and forms here the secretory component and secretory immunoglobulin A. This secretory component and secretory immunoglobulin A, sorry, should I pointer? Okay. This secretory component and secretory immunoglobulin A then binds with the foreign pathogen, like it tag that, and then macrophages comes and phagocyte that those foreign pathogen. But here, this pneumonia uses our own host machinery against bacteria for its invasion. What does it? What it does, that it binds with this PIGR, and by the same mechanism, transcytosis, it invades our body. So let's see how it's interact with the PIGR. So in pneumonia, it has a surface protein called PSPC or CBPA. PSPC is pneumococcal surface protein C, and there's CBP also choline binding protein A. Both are same, but different scientists named it as different way. So in PSPC, it has 4 domains, that is FN binding domain, R1 domain, R2 domain, and these are repetitive choline. So these bacteria, R domain, it I should go the previous slide. This R domain binds with this PIGR, and this invades to our body. Let's see how it binds. Okay, here is the these CBPs R domains interact with the PIGR. So it's like a PIGR is our cellular door. So it's like it has a key, that is the CBP R domain. So it uses this key to open the cellular door PIGR and invades our body. So we thought if we stall the interaction here, then we can stall the invasion. So for that thing, we think that we can make a protein inhibitor. So here is our solution. For making that protein inhibitor, we have taken a scaffold protein, that is adnectin. So why we have taken scaffold protein adnectin? It is small sized protein, and it has a CDR loop like structure, that is so CDR loops are found in antibody, and here you can see the loop, FG loop, BC loop, and DU loop. The special feature of this protein is that if we randomize or do some mutation in these loops, it will have no impact on its overall stability. And it is a small size protein, nearly 15 kDa, but it has special abilities like monoclonal antibodies, which are nearly 150 kDa proteins. So due to its lower size, small size, it can bind with high, it has high penetration power, and it can bind with its target with high affinity. And also for antibody production, we need cell culture, but for this adnectin production, we can also do it in bacterial cell. We don't need mammalian cell culture. So it is highly cost effective. So let's see how we have designed this adnectin to our ADR. So for designing the ADR, we have first see where these PIGR and CBPA interact. This is the D4 and D3 domain of CBPA. It interacts with the PIGRs. Sorry, D3, D4 domain of the PIGR, and it interacts with the CBPA. So we have just mimicked the environment in this while designing this ADR. Let's see how we have done it. So one here I have shown the C alpha atoms of PIGR interacting with the CBPA. So we have just grafted these residues, interacting residues, to the adenine and made a novel protein, that is ADR. And we have here superimposed the all that three proteins, that is PIGR, adenine, and CBPA. So here you can see CBPA is interacting with PIGR, and we have grafted these residues rationally, because we have done this in rational protein engineering in the adenine and made ADR. So here is just the C alpha atoms of PIGR and CBPA interacting, and we have grafted those residues in DE, BC, and FG loop. So here you can see, here is the ADN, the base scaffold molecule, and our ADR is also here. This ADR is the blue color. You can see the red one is the base protein molecule, that is adenine, and our designed molecule is this one, ADR. So it is almost similar, but due to some randomization in the loop, it is because we wanted to, we wanted it should bind with the PIJ, that cellular sorry, it should bind with the CBPA. So for after modeling, we checked computational checked its stability. So for that thing, we have done a molecular dynamics simulation. Here is I am showing the RMSD and RMSF. So why RMSD is needed here? So RMSD is the root mean square deviation. It shows the molecule's deviation from its initial conformation. Though here we are not getting plateau, but it is increasing. But I have taken also this result because here ADN and ADR both are showing same trends. You can see here, and here in the RMSF fluctuation, there here some residues of ADR showing high fluctuation, because we have randomized the loop, and these loops are showing very high fluctuation. So let's see what is this RMSF. Unlike RMSD, where RMSD shows the deviation of the whole structure, but here in RMSF, it shows the deviation of individual amino acid residues. So this high fluctuation is caused because of the randomized loops. After that, we have checked the radius of gyration. So why we have checked this radius of gyration? The reason behind assessment is that this radius of gyration shows us the compactness of the molecule. So here you can see that our base molecule is more compact than our design molecule, but the trend shows it is quite stable. So why this compactness is important? If the compactness is very high, that means our protein is very stable and it is not going for denaturation. And here you can see the protein is computationally showing very promising result, and it is very stable here. So after that, we have checked the solvent accessible surface, the SASA value. That this parameter also showed us that our both the molecules, in spite of randomizing the loops, both the molecules are more or like very similar in stability. So after getting the promising result in stability of ADR, we have docked this ADR into the CBPA R2 domain. So this also showed us a very positive result, that it shows binding energy binding score of more than less than -63. That is very good. And here is our interacting residues. You can see. So to check the stability of the complex, we have done MD simulation, and throughout the MD simulation, the RMST value, RMST graph is showing that it is quite stable. Here you can see it is more stable than the individual ADR. You can show here in individual ADR and adnectin, the RMST was going up. That means it for the for equilibration it needs more time. We have done simulation for only 200 nanosecond, but in complex it was more stable. That was a very positive result. Here I have shown the RMSF value. Then RMSF also, the both you can see here the fluctuation is low than the individual protein molecule. So this also shows this dot structure is quite stable. So here is the radius of gyration. Here you can also see both the protein molecule in complex is highly compact. So after getting this type of positive result, we have gone for the synthesis of our designed molecule. For that thing, we have used bacterial expression system. We have our ADR molecule, we have cloned in PET28A vector. Then we have transformed it in E. coli BL21 bacteria. So after that, we have optimized the protein expression. And after optimizing protein expression, we have purified it using nickel NTA affinity chromatography. For that, we have first cell harvested by centrifugation, then lysed the cell by sonication, and then we have collected the supernatant, and this supernatant we have run on the Acta Pure instrument for affinity chromatography. We have done by using nickel NTA column. After that, we have for extra purity, we have done gel filtration chromatography. So how we have done nickel NTA, I want to show you. For the nickel NTA chromatography, our protein is engineered protein, and we had already put his tag there. This his tag binds to the nickel beads. So first, this is our workflow. First, we have equilibrated the ACTA system with our lysis buffer. Then for the binding, we have pour our protein into the column, and that protein which contain his tag, that is our protein of interest that binds to the column, and then we have washed that. After that, we have done elution by using elution buffer that contains imidazole. So after isocratic, we have increased isocritically, we have increased the imidazole concentration, and that eluted our histact protein. After getting the histact protein, we have done tape cleavage using tape protease. For that thing, here we have collected the eluted protein for getting the ADR with histact. But after that, we have cleaved the our protein with tape protease, and then also done the similar nickel NT affinity chromatography. There, this histag binds to the nickel NTA column, and in our flow through, we have collected our protein. After that, we have done the gel filtration chromatography. So, how this gel filtration chromatography works? So here you can see it mainly works on the size of the protein and the shape of the protein. So by gel filtration chromatography, we have got our very pure protein that is cleaved ADR, and then after getting both the protein, we have gone for the binding studies. Here you can see here, and we have done binding studies by using MST, that is molecular scale thermophoresis, and that shows us a 9 kd value of 9.94. So 9.94 in micromolar. So we have got a protein which has a binding affinity in micromolar. That is quite good. For our therapeutic antibody, we use proteins in nanomolar affinity, but here I have got micromolar. So further I am doing optimization for better affinity. So from this study, what we have got a binder against CBPA, which can halt the protein-protein interaction of CBPA and PIGA. So it can be a therapeutic inhibitor, and we have cloned and expressed it from bacterial expression system. So the production cost is very low compared to any antibody. So we are still further doing cell-based study for for further validation. So this was my research on novel inhibitor binding. Thank you. Much time takes. Okay. So do we have any questions?
If anybody has any question, you can write it in the chat. So the speaker can answer.
Hi, Arkadada.
Hi.
Can you hear me?
Yes, yes.
Yeah, I am Shivam. Actually, I have a small question to you. When you were doing that Guy rating and compactness of those thing, have you
Yes.
done any previous studies data? Have you looked at it?
This ADR previous study means what?
Designing.
This ADR, okay. This ADR we have designed for the first time, and this base molecule adnectin is also first time we have used for targeting this CBPA protein. So I did not get any type of similar data previously. So
Okay, and in general question, have you because you might have gone through, I also have this, what is the relevant compactness you are looking for to consider this molecule can be considered
yes.
for further like cloning or whatever expression studies or whatever or synthesis. How you take that call when you're designing from in silico to this thing?
Okay. So here what I have done. 1 minute. Let's show the slide.
Mhm.
So adnectin was the base molecule that is therapeutically used for other studies. So what I have done, I have relatively checked between the results of adnectin and our designed molecule. So here, what I have found that both are almost similar. That's why I have gone for further studies.
Okay, but like as a layman, I'm asking, is there any, what is that, biological reference or any previous study references when from in silico to when you're moving into actual any expression studies or whatever it may be, or biological things? Is there any, what is that kind of I know it is a little difficult to come to a conclusion, but just out of curiosity, I'm asking: is there any, what is that? Yes, if it is this much, we can go ahead. This will be relevant for any other systems also. Is there any such kind of studies?
Okay, so you are asking from where we are here we are getting something promising result that we have gone through uh, synthesis of the protein molecule. So,
Yeah,
so are
because
you asking
most
that?
of the time, I
Yes.
whatever
Okay.
is designing and all those things. By this time, we could
Okay.
have all the what is that troubling proteins as a treatment options. We could have been
Mm.
answered all those things. But there should also be something what is that boiling point that which is not answering from in silico studies and in vivo studies are not matching with each other. But because you are also what is it doing in this domain research, just a
Mm.
curiosity point I'm asking, is there
Okay.
any mandate things or guidelines or something like that? I'm not just telling what is that standard guidelines as an
Yes.
industry level or expression study, because from in silico to in vivo, when we move or whatever, how exactly you take a call, this compactness would be a right, this gyration is would be a right or whatever. How exactly you take that call? Just not only this one I am telling you,
Okay. Okay.
I'm
Okay.
asking you in general, is there any such kind of things in this from in silico to when we move into biological expressions?
So, so I have not this much. Previously I have not thought about that. That what I have in this case, I have just seen that both are like similar. So go further, and the docking result was also very good. But for your question, I need to check that you are asking that how much. So your question is how radius of gyration is how much important for going computational to uh, cell-based in vitro studies or
Yes.
something.
Yeah. Yeah. Whatever, whether in vitro or in what is that cell-based studies or whatever it may be, when you
Yeah.
consider in silico designing to move into what is that cell culture or yeast expressions or animals
Okay.
culture, is there any what is that standard this much from this much to this much? These are all the things we need to consider. ऐसा कुछ
Yes,
है
for
क्या?
that thing, what I have done, we have not taken this radius of gyration that much importance. Our main goal was that we should design a protein that should bind with that uh, CBPA. For that thing, what
Mhm.
I have done, we have here checked radius of gyration to see the compactness is very much different from our base molecule, that is adenectin, or not.
Mhm.
So
Mhm.
here we have
Okay.
got it is quite similar, but for
Okay.
going to the bacterial expression, we have first docked this ADR with the CBP-R2 domain. We have done it using haddock, and there we got a score of minus 63.8. And
Okay.
that thing, why I have taken that, because when people works on some drug, here their binding scores are quite not that much good, because drugs are relatively small than this protein molecule. But
Okay. Okay.
uh, what I have seen, when we someone design a protein inhibitor that is showing more
Mhm.
than minus 40, so that was I need to check all those things again, but
Mhm.
that score was very good. And I think the Beirut surface area of uh, this interacting two protein was nearly 766 angstrom.
Okay.
So
And also, okay.
yes,
For
So,
this designing, what softwares did
um,
you use, sir?
yes, I have used Swiss model.
Swiss model. Okay.
Yes, yes.
Okay. 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 Thank you.
Because Swiss model also uses the homology designing, and we had that this adenectin as a base model, and we had also crystal structure of this adenectin.
Okay. Thank you.
Thank you.
so there are some of the questions in the chat also. So let me tell you that. So one is from the Surendran, and one is from the Chaitanya Kushal Tukle. So first I'm going with the question from Chaitanya because it's a more of a specific thing with this topic. Then we can go to that of the Surendran. So the question is how to prove the simulation study in biological systems, which is how in silico drug docking and results correlate with the biological experiment result.
Okay. For that thing, we have to synthesize how in silico drug docking results correlates with the biological experiments results. So for that thing, you need to just in our lab we also design drug, but for that thing, what I have seen, they first synthesize the drug. If they got some promising result in silico, then they synthesize the drug and then do in vitro studies. For that thing, you have to first synthesize drug and then you can do.
So one question, the question from Surendran is that what will be the next step in your study and what are you planning to do next?
So next, so it's a therapeutic protein. So first we need to do the in vitro studies. For that we need to do in vitro cell-based adhesion and invasion assays. So in presence of, we need to see in presence of ADR, is it really um, stalling the bacteria from invading a cell? Because here we are seeing just a binding, so it is binding, but in a cell how it is showing its affinity, and we also see its immunogenicity and toxicity. This type of studies we should do first, we have to do. And after that, I have also checked this protein is very thermostable. So if we can give it as in a spray, we can also do, because here this inhibitor is not killing the bacteria, it's just stopping the bacteria from invasion.
Oh, so there is, I think, Jannavi. She wants to ask some questions. So Jannavi, you can ask your question.
Yes, hello, am I audible?
Yes, yes.
Yeah.
Yes, you are audible.
So
You can.
that was a yeah. So that was a very interesting talk. Thank you very much. My question is that you studied the radius of gyration. If I'm wrong, please correct me. You studied the radius of gyration of the protein before you went for the protein production and the further in vitro approaches from the in silico approaches. So could you please explain to me again the relevance of knowing the concept of protein
Mhm.
gyration and the relevance of the concept in the stability of the protein?
Okay, okay. So let me. So if throughout the simulation, if radius of gyration is increasing, that means the uh, protein compactness is less. So if the protein is so that means protein may be going for denaturation or conformation change. And what I have done here, I have taken radius of gyration because I want to see if the while we have changed some residues in the loops region, if that is making our design molecule unstable or not from the base model, that is adnectin. And we have not gone for the synthesis after this ADR simulation, ADR and adnectin simulation. We have gone after the simulation of the both complex of ADR and CBPA protein, because first we checked this we have done ADR and adnectin simulation to check how much ADR is different from the adnectin. We know that adnectin is quite stable. If, so after engineering adnectin, we have made ADR. We want to check how much this ADR is also stable or not. So when we got some promising result that ADR is quite stable, here we are seeing that compactness is not that much, deviation is not here. So we have after getting this result, we have gone for the docking, uh, and we have gone for bacterial synthesis after the docking result. So am I able to
Yeah.
answer your question? Or
Yeah,
something else?
that answers my question. And I have, excuse me, I have a follow-up question that if this radius of gyration results wouldn't have been satisfactory, that would have indicated that new molecule you want to synthesize, the protein peptide, is unstable. So how would you have
So for the
changed
protein
the peptide
instability,
to solve the
okay,
issue?
okay. So for that thing, we have done the only this radius of gyration does not define the stability of the protein. For that thing, we have also taken several other parameters. Those are RMSD, RMSF. So here in RMSF, it is showing these residues are showing very much fluctuations here. So we have first gone through these residues then, and here we also have not done the full simulation here, because here you can see it is going up this RMST. It is not equilibrated yet, but we have also taken this result because we have seen that this ADR and actin is showing similar kind of trends. So if there was not satisfactory result here in radius of gyration, we have then checked this RMSF and RMST, and this RMSF will all tell us that which residues are behind the stability of that instability of that protein.
So what you mean is that if the RMSD values and the RMSF values weren't satisfactory, from that
So
you would have also understood what is causing the, like what residues exactly are causing the issue. You would have changed those residues.
um, I do not have
Um,
changed. Then we have to think another way, because we have changed residues to such a way that it should bind with the CBPA. But if
yes.
those residues we have to change for the stability, then that then we could not do that. Then we have to design again. We have to take another base molecule then.
Hmm.
Uh,
Okay.
or
Yeah,
else
that answers my question. Thank you so
yes.
much.
Is there any other question?
So in the meanwhile, I also want to ask you one question. So uh, AIIMS, it is a premier medical institute in the country. So can you just elaborate on what are the research opportunities in AIIMS, or what are the topics that they work on, things like that?
So in our biophysics, our department mainly works on the structural studies like crystallography, cryo-EM, and there are one two labs which also works on the proteomics. But our lab we works on the um, protein engineering, drug discovery, and also RNA works. There are some RNA works. And in biochemistry, what I have seen, there is also they works on CRISPR-Cas9, and I need to check, but I have told what are the major works in our biophysics.
That's nice. So does anybody else have any questions? You all can pitch in any questions of yours. So Arkadyuti, I think everybody technology asked the questions. And okay, there's one more question. Anushka, she wants to ask.
Okay.
Is that paper? You can go.
Hello. Yeah.
Yes.
So actually, I had this confusion that is there any summer internship program in this like AIIMS Delhi because I'm a nanoscience student.
Okay, so there is not like specific summer internship. You can talk to PI
Approach the-
anytime. You can mail anytime. And here I have seen some students. Yes, there is here PIs are also interested in taking intern.
Okay.
So
And
you
like
have to
from
just
which
mail.
website can we see that?
You can take the mail ID from AIIMS website.
Okay, okay, thank you.
Yes.
So it was very interactive and very nice session from you. Thank you for that. And a lot of people have different from the videos that it went to the beginning. So it was also good for us to listen to something else that we're doing. And I hope you have also enjoyed it. Thank you for the coming. And so now maybe you can close the session. If you have anything to say, you can also say before it. Any comments or anything?
Thank you to all.
So we are ending the session here today. Thank you for attending the session.